So, welcome this evening to all of our members who are joining us for our latest Chat with a Scientist. We are so glad you're able to join us this evening. We have a wonderful couple of speakers here with us tonight to talk all about gene editing, something I am excited to learn a lot more about. Tonight is all about answering your questions. Our panelists will each do a short introduction of themselves. We'll ask a couple questions to start, and then we'll have the floor open to your questions. If you want to send a question to our speakers, there's a Q&A box at the bottom of the page here on your screen. Type in your question, hit submit, and it will go to our moderators who will then go through those and read them to our presenters tonight. Additionally, this webinar is being recorded and it will be shared at a later date, so be on the lookout for that if there's someone you wish to share it with who was not able to attend this evening. Tonight, we're joined by two scientists who specialize in the field of gene editing. We have with us Dr. Andy Ravanelli, head of cell and molecular biology at MilliporeSigma, and Dr. Casey Jowdy, senior product manager, gene editing at Agilent Technologies. Andy, do you want to do a little more lengthy introduction of yourself? Oh, I think you'll have to unmute.
Fail right off the bat. Thank you, Christina. Thank you, everybody, for having us. My name is Andy Ravanelli. I'm a St. Louis native. I did my undergraduate work in genetics at Southern Illinois University, Edwardsville, and then moved out to North Carolina and did my doctoral work at Duke University in cell biology, and then did a fellowship in pediatric oncology at the University of Colorado Hospital School of Medicine. And my backgrounds have been genetics, neuroscience, and developmental biology. For the last 7 years, I've been with MilliporeSigma, Sigma Aldrich, and then MilliporeSigma, and in the genome editing group as a principal scientist and senior scientist in genome editing. And now I'm the head of cell and molecular biology at MilliporeSigma.
Wonderful. And Casey, do you want to give a little lengthier introduction for yourself?
Yeah, absolutely. So I'm originally from Arizona. I did my undergrad at Grand Canyon University in biology, a minor in chemistry. From there, I went on to the Ohio State University in the Department of Molecular Genetics. And I studied how the early nervous system is wired during embryogenesis and what genes are involved to allow those very intricate neuronal circuits in Drosophila as a model system. So from there, I went on to Washington University in St. Louis, which is what brought me here to St. Louis, where I did work in the dilated cardiomyopathy lab, where we would take genes that were involved in dilated cardiomyopathy from genome-wide studies, and then model them with different genetic tools in mice, in cell culture, and Drosophila, and try to understand better treatments for those rare genetic disorders that contribute to dilated cardiomyopathy. And then from there, I started at a small startup company in St. Louis called Cofactor Genomics. And there, that is a next-gen sequencing company. I think this will come up a little bit later, where you actually read the genome. So that's an important part of gene editing, is to be able to read the genome if you ever wanted to edit it. And then after my time at Cofactor Genomics, I worked at Sigma Aldrich, MilliporeSigma, where I was a colleague of Andrew Ravanelli's, and we've been friends ever since. And now currently I'm at Agilent Technologies. I'm senior product manager there, and I continue to do gene editing and DNA synthesis work and help enable researchers around the world do gene editing.
Awesome. Well, thank you both again for joining us. So excited. I'm going to start with kind of a broad question. What really is gene editing? We hear this phrase a lot. What does it mean?
I can probably take that at the start. So gene editing is the process of directly manipulating the genome, the sequence of the genome or the function of the genome in cells or in an animal. There are several different technologies that have been developed over the years, over the last 20 years or so, that continue to improve the way we can access specific sequences or target specific regions of the genome to affect a change on their sequence or their function. That started out with zinc finger nucleases, and then the technology transitioned to a slightly newer technology called TALENs, and then finally into the CRISPR era, as we call it, where each protein got successively smaller and more specific. We were able to affect more control over the sequence of the genome. The process of gene editing requires the engineering of a protein or an RNA, a very small molecule that needs to enter the cell, enter the nucleus, bind to and act on the genome itself. Usually it results in, or it causes, a double-stranded break in the DNA. So CRISPR, the gene editing tools that I've talked about are often called molecular scissors. They'll cut the DNA. The cell will undergo a programmed repair response where it will repair that double-stranded break. And we hope, and usually the way we plan for it is that the repair results in a sequence change to the genome in the way that we want. Oftentimes the protein or the targeting complex is removed from the cell. It degrades quickly over time. It does its job and degrades if you don't keep adding more of it. So it enters the cell, does its job, and then degrades right away. And then that cell is then permanently altered for the rest of that cell's life and the progeny of that cell in a sequence-specific, identifiable manner. And then ideally that results in a change in how that cell behaves. If we are looking to study how a gene functions, first thing we do is try to break that gene and see what problems happen. So in cell culture or in animals, we'll use gene editing to create mutations that cause a gene to malfunction. For clinical purposes, the goal is to identify a change using sequencing, the way Casey has described it, and then do that same targeting with gene editing to go in and correct a problem in the genome, often a single base or a single letter that's been mutated either through random chance or through insult to the genome, and then correct it so that the cell behaves appropriately from there on out.
Well, that's great. Casey, do you have anything to add to that or any other thoughts?
No, I think that's a really good kind of summary of the history of gene editing and the tools we can use. I think I'd just break it down into a couple different patterns we can think about to kind of give us a hook on the power and potential of gene editing. And one, gene editing allows us to unlock all the mysteries, or hopefully all the mysteries of the genome and how it can function. Like there's 20,000 genes in the human genome. And what do they all do? So there's a very kind of basic question about those genes. And then as we learn more about those genes and if they're involved in a disease process, with CRISPR and the other gene editing technologies, we may be able to have the power to correct them. And I think there's a couple different ways, like an analogy that really helps me is think of gene editing in a way where, we can read it first. And then if we want to make an edit, there's a way where we can knock it out to remove that function of the gene, almost like a light switch. We get when it's on, we can see the light, and then when we can turn it off, we can see what happens when that gene's no longer functioning. And then there's another aspect of CRISPR technology where it's almost like a rheostat, where you can kind of dim the lights a little bit and turn it way down, or you can kind of go medium and then higher levels to see how that functions. And that's really relevant for cancers and other diseases that are out there that we want to, potentially treat. But also, I think what's missing from a lot of the public eye and what's talked about, like on YouTube videos and even on late shows, I think the Late Show or I can't remember his name now, I'm forgetting, but anyway, on public TV, and is the fact that you can study the entire genome. And I think only about 5,000 genes are kind of known what they function as. Well, what do the rest of the 15,000 do? And I think with the potential to use gene editing in a much more global manner will help us refine, what the genome does to potentially unlock other therapies or understand who we are.
That is astounding. I did not realize that. That's crazy. So you both mentioned CRISPR several times. What exactly is that? And what should we know about it? Why is it important? Hear that a lot.
Do you may go, Andy, and then follow up? Yeah, I think CRISPR in a simple sense, and in a way that's kind of a good analogy, is I think you could think of as a word processor, but I also think it's molecular scissors, so it could go to a very specific spot within the genome, exactly where you want it, and it can make edits there in a couple different ways. One is as scissors, but you can do so many other things with CRISPR, and it acts like a tugboat, if you will. So if you have an address in the genome that you want to go to, so CRISPR can go to that location. It can make a double-stranded break using molecular scissors at that point in DNA, and then you could do a lot of different things with those, hopefully with those breaks where you can select them. And the other thing is you can add modulators, like I said, that rheostat aspect of CRISPR, where we can slow genes down and then also speed them up in a way. So I think that's kind of how I would approach that.
Challenge that gene editing has faced in, let's say, mammalian cells, or the challenge that the field of genetics and repair has faced, is that there's more than 6 billion letters or nucleic acid bases that make up the 23 pairs of chromosomes in a human cell. It's the instructions for all of life for that cell. It contains all of the information that that cell will need. CRISPR targets, is able to go in a sequence-specific manner and find 19 to 20, use 19 or 20 bases or nucleotides or letters to find the exact spot in the genome that matches that sequence, so that we can direct whatever we want to do with CRISPR, whether it's making a double-stranded break, making a cut like scissors, making a nick, which is only a single-stranded break. The genome, human DNA, is a double-stranded helix, as we've seen with pictures. To be able to affect the change that we want on the genome, whether it's to make a cut or deliver some cargo, another cargo protein or another molecule to that spot in the genome to turn a gene on or off or up or down, requires an inordinate amount, an almost fictional amount of specificity. And CRISPR allows us to do that. So CRISPR is the latest gene editing technology, and it's been around for the last decade, which is a pretty new technology, and it's come a very long way in the last decade. The power of CRISPR to actually target the places and do the things that we want in the genome has already led to an exponential increase in the number of publications using it as a tool, all the way into clinical trials, which is fast for a new technology to be discovered, made, turned into a functional tool that's somewhat democratized genetics around the world, all the way into clinical trials and therapeutics in just 10 years is pretty astounding, and it illustrates the power of this tool. And that's what it is. It's a tool. Gene editing is a field, and CRISPR is one of the tools that we use to edit genes.
Oh, awesome.
Can I just add
yeah, go ahead.
This comes from a little more historical perspective of what CRISPR is. It's actually a bacterial immunity, and the CRISPR system comes from bacteria, and that allows them to fight against phage and other bacteria. And it has a really interesting history. It almost seems like a complete accident, but it really goes back to basic research and then all of these different fields coming together in order to enable this. And so I think the first observations of these CRISPR, which is a clustered regular interspersed short palindromic repeat, that was found in bacteria when they started these gene sequencing projects. And so that led to, so this is 1987 and took all the way to 2012 for this, all these different fields. There are biodefense, yogurt culture scientists, Yoplait was involved in this, and all these different scientists. And finally, Jennifer Doudna and Emmanuel Charpentier were the ones that kind of put it all together. But there were so many people involved in the discovery and putting all the pieces together. I think it's fascinating to kind of look at that aspect of it, like how it all came together. It's incredible.
That is so cool. I did not know about that either. So I will have to do more research and learn even more, because that sounds like an awesome story.
A lot of dedicated biochemists, a lot of dedicated time spent doing basic biochemistry, basic protein characterization and analysis before it was converted from just a thing that bacteria were found to do as part of their adaptive immune system, if you will, to being turned into a tool that could be used outside of bacteria to do real work.
Wow, that is so cool. We had a question come in from the audience, and they were looking for, can you please give a concrete example of a success in gene editing? And they said an example of maybe something that didn't go so well with it, just anywhere along the history.
Yeah. Andy, you want to go? Or I mean, yeah, I have some ideas, some thoughts on that.
That's a very good but very broad question to ask. I've had successes in my lab with CRISPR. I would say getting CRISPR, let's say the first big CRISPR as a gene editing tool success came in its ability to cut DNA outside of a bacterial cell. So being able to just purify the protein and use it to cut DNA. And then a big leap came when it was used by my colleagues to be able to put it into an animal cell, and then by kind of good luck, it was able to we were able to get it inside the cell, but that it went into the nucleus, which is another level of difficulty that this has to overcome, and find its spot in a complete intact human genome in a live human cell and make a change. That was the first big, this is going to work in animals, this can work in humans, this can work in people, success. And then all the way to now, if just a few years ago, the first human clinical trial started in 2019, using CRISPR to correct single gene, single point mutation mutations in patients with beta thalassemia and sickle cell disease. Those are both diseases that are caused by a single mutation in a single gene that causes significant clinical complication, blood disorder. And to be able to take human cells out of a human, out of a patient, make the change to correct that mutation, put those cells back into that person, that same person, so that we don't have to undergo potential graft versus host disease. And then for that person to have restored blood function and still be healthy, safe, and for the most part disease-free today. That's a major, major success story.
That's incredible.
Beat that, Casey.
What's that?
Said beat that, Casey.
Well,
What are some other success stories?
yeah. I could think of, okay, so I could give a example of a success story and a potential disaster in gene editing. But let me start with the sentimental story. So the question asks a concrete example. This is a personal concrete example that I witnessed at my first conference that gave me the let it be like, this is not just on paper, this is not just in theory, this is going to work and affect people's lives. But it was early on, and so there's a lot of complications. In one sense, gene editing is very easy on paper, right? Or it seems easy, but delivering it and making sure that edit delivers where it's supposed to, not anywhere else in the genome, is the challenge. And we have to overcome all those things in order to be successful. So that's one. So this, my story is better because it's about puppies. And in this case, I went to a conference, and I won't mention the company or the researchers, but they're studying Duchenne's muscular dystrophy. And it happens to be, it's very sad that the research didn't cause the muscular dystrophy and the beagle puppies, but they were born with it. And they wanted to see if gene editing could help restore the muscles strength it mostly affects the hindquarters. And so they have a video of all these cute little puppies running down the hallway, and their little litter mate who had the Duchenne's muscular dystrophy, couldn't, because he didn't have the strength. So he kind of hobbled along and wanted to get the treat. And so they would all jump up and get a treat. And he wasn't able to, but they still gave him a treat. And then they were able to introduce gene editing into the muscles, specifically into this puppy. And 4 to 6 weeks later, he had recovered a lot of strength significantly in order for him to be able to keep up with all his other litter mates and hop up and get the treat from the researchers there. So I think that shows me that this can be successful and impact the lives of people. And I do have friends that have their son has Duchenne's muscular dystrophy, and I hope someday that we can get to that point where that's able to restore a lot of the muscle loss that he's endured and affects his life every day. So that's, I think, a success story, and I saw it with my own eyes, and I have close friends that are impacted by it. And the other one, I think, an example of a disaster, and I think that's ethically, it could become a disaster. And I think we need to learn from the past, and specifically in eugenics. I think that could be a huge problem. We have to be engaged in that, because even if we could, should we? And I think those are the important debates that need to happen in the public square for policy, because it's a very powerful tool, and we need to ask those critical questions. And I think that could be an example of a disaster. And so I think it's very important for everybody to be involved and engaged in that at every level, whether you're a scientist or not. So.
Yeah, and there have been a lot of conferences and symposia drawing up globally accepted or nationally accepted ethical guidelines for how gene editing is to proceed, and what's appropriate, what's viable, what's appropriate. I mentioned earlier that there are 6.4 billion bases in all of the chromosomes in a human cell, and that only 19 or 20 of those are needed for targeting CRISPR in one case to a specific spot in the genome. That means that it has to be very, very specific. And one of the challenges that we've had to overcome is modifying the protein, modifying CRISPR, improving it to improve that specificity, because there are obviously many, many opportunities in the genome where it can target the wrong place. And if you send this scissors to cut the wrong place in the genome, you can cause other complications, other genetic consequences we call off-target effects that can significantly or not alter the biology of the cell in ways that are unintended. Early attempts at gene therapy there were examples of lethal consequences early on in the clinical trials. At the time, those gene therapies were not necessarily rushed, but there were a lot of consequences to it that should have been addressed or explored earlier in the lifespan of that technology. With CRISPR, the technology works a lot better. It's a lot more reliable, but the field has been taking it much more slowly, much more seriously as to calculating and anticipating what are all of the possible consequences to the genome that this could have outside of the very specific change that you want to make. And so choosing where in the gene, choosing where in the genome to make these targets, which genes to target, which types of mutations, which types of organs we can successfully get this in to affect a change. Because a lack of success early on in a clinical trial in a particular technology can damn a trial or that technology's future very easily. And so a lot of care has been taken and continues to be taken to treat this with the respect that it deserves of trying to bring a technology into a human person for a clinical trial or for a clinical therapeutic. That level of care exists in the ongoing approved clinical trials, but there have been cases, or at least one very public case, of a scientist in another country pushing this technology forward into humans before it was ready, without taking care to, without giving it the care that was necessary to predict what potential side effects or off-target effects could happen. Two girls have been born, twins, who have had their genomes edited, and with a real lack of transparency as to the planning for this, with a real lack of transparency to even the parents, and against the ethical codes of the institutions and the countries and the legal complications of performing this kind of gene editing on embryos that will result in patients or individuals being born who had no choice in this process, and who may have genetic and lifelong physiological consequences to that process. It was done, we hope and we assume, in good faith to help these individuals. It was regarding trying to create an immunity to HIV, where HIV existed in this family, but it was considered somewhat ethically disastrous to the field of gene editing. Yeah.
That ties right into a question that actually we had come up, which says, my 7-year-old is very interested in understanding how or if scientists might use gene editing in embryos. I think you answered this a bit. What diseases are scientists looking at for using this method?
Yeah,
there are a lot of diseases. Embryos is a hard thing. So my background is in developmental biology and birth defects. The problem is that many of the birth defects that get the most attention, or that have the most that are the most outwardly visible or clinically significant, often occur in the embryo before the mother even knows that they're pregnant. So those genetic consequences happen right away and set that embryo on a course for a birth defect or a genetic defect too early on to be corrected. So the ability to then go in and correct that becomes severely limited in a way that will be meaningful for that child once they're born. gene editing in embryos in, say, an in vitro fertilized embryo is definitely a possibility. The ethical, it's been done. It's been done in people. The ethical implications of that are that you are taking away, and ethics can be, sometimes there isn't a right or wrong answer. It's very context dependent. But some people will argue that modifying the genome of an embryo or a mother's eggs or a father's sperm will result in a person who a very specific decision was made for them about their future, their genetic makeup in a way that they did not have a choice over. That can be argued either way. I'm sure that there are people who have significant genetic anomalies in their family, like Huntington's disorder or Huntington's disease for one, devastating neurological disease that's 100% lethal. Given the opportunity to correct that in your family lineage would be viewed often as a good thing. But if you do that, then you do take away that choice. So that's a ethics often are in the eye of the beholder. So someone who may be, we may all agree that Huntington's is something that we would want to correct or change. But if you wanted to affect a change that, say you have myopia in your family. Myopia is short-sightedness or a vision disorder, but it's also can be correlated with intelligence. And so making a decision to correct a person's genome in a way that you unilaterally, the decider, decides is worth correcting may not be something that someone else wants corrected in their family. Communities that have genetic deafness or blindness sometimes have a strong cultural meaning to that particular trait in their family. And so while an outsider may deem that as a disease or a mutation that needs to be corrected, that decision may be better left to the family themselves or to the individual that's going to be impacted. So major moral and ethical implications to taking away choice.
That's great. Casey, do you want to add anything to any of that?
Yeah, no, I think we addressed it earlier. I think that is a potential for disaster in terms of, yes, we could do it. Should we do it? And in terms of, how, yeah, those tools are there for us now. And in terms of editing embryos, we need to have strong ethical standards. We need to ask where those standards come from in order to be able to set forth policies to protect the embryo, that life that's going to be affected without their choice. And also, what are their goals? So, I mean, I think we're still a long way in order to be able to do that. If we do that, so I think, and also knowing at any point, you're not going to know the outcome because some genes only affect certain tissues like Duchenne's muscular dystrophy. It only affects the muscles. So, but when you go in and you change the genome, that goes down and it goes down to their children. So it not only affects the individual that had undergone gene editing, it's now going to be passed down, from generation to generation. So it doesn't just impact that one person. It's not an isolated case. It's going to affect a lot of people. So I hope, and I think a lot of laws have come down, putting in a moratorium on the types of things that have been done without oversight and everything. And since we're a global world now and connected in so many ways, I hope that that gets spread and the right ethical standards are put in place globally, worldwide, that affects everyone.
It's heritable genome editing is illegal in the United States. There's no funding for that, and it's illegal. There are some diseases that you could only correct very, very early on before choice is available. Some diseases, you have to be able to access the tissue when some diseases are very hard to correct because it's very difficult to get. You have to deliver that protein some way and the RNA that's associated with it with CRISPR into the cells. Blood disorders are a great first place to start because it's a liquid organ. So you can get it, you can take it out of the body, you can manipulate those cells and put the cells that it works right back into the body. Or you can inject it into the bloodstream and it will go to most of those cells or many of those cells. Muscular dystrophy, much harder. The brain, much harder. Solid organs are very difficult often to get enough cells functioning, to get delivery into enough cells to change the function of the cells in that tissue. So we start with the cells, we start with the diseases that we can actually make hay on. And so the earlier you get in an embryo, sometimes the easier it is to get those future tissues corrected. But then there's more risk associated with it.
Great. Well, thank you for that insight. It sounds like there are a lot of things ethically, technologically, all kinds of stuff taking place with gene editing. How do we, as just members of the public, kind of stay on top of that? One of you had mentioned that we should be informed, as citizens, and as things happen, we should be informed as well. So what's a good way for us to stay kind of informed about this information? Casey, do you have any thoughts?
That's a great question. Yeah, it's a great question. I mean, part of it's just being engaged, right? In terms of finding out more about it, there's so much public information out there through the internet, YouTube. It makes it to public talk shows. It makes it to movies, popular movies. Some of them egregious in terms of what they're doing and kind of the overhyped, science fiction aspect of it. And then policies too. I think those are important, and medical treatment, HIPAA laws, all of those things, they're going to affect you. And I think you need to be aware of what's going on in the world and, how it impacts the healthcare system overall, and then, society as a whole. So I think being engaged, coming to stuff like this, being curious.
I would say any opportunity that you have to talk to a scientist, to ask a scientist directly, or to get the information from a reputable scientific source. If you want to know about CRISPR, and if you want to know what's going on with CRISPR, Facebook is probably not the place where you're going to go to get that information. Your news talk shows, probably not the best place. Primetime television, sitcoms, definitely not the best place to get any scientific information at all, really. The National Institutes of Health, NIH.gov, is a great source for all, as a great repository for science just in general. But they also have a lot that makes it very accessible. The American Academy for the Advancement of Science has a nice website that can be sometimes a little deep, a little too many details, depending on the type of the literature that you're looking at. But your local universities and hospitals will also have, or medical universities will often have a great website. The Innovative Institute, the Innovative Genomics Institute, the IGI, innovativegenomics.org, I believe, is at the University of California, Berkeley, and University of California, San Francisco joint venture. And they have a great website that really breaks down all of the clinical trials and successes in a very accessible format. So anytime you have questions about science, what's happening in science, go to a reputable scientific source, always.
Great. I had a couple of other questions that have come in. One was on, so you'd given Casey the example of these dogs that had the gene therapy. Questions kind of on that specifically, but I think broadly, if a gene therapy is used, does it need to be repeated all the time, or is it kind of a one-time thing and then it's done?
Yeah, great question. And it's context dependent. So for cells that have a high turnover, like in the blood lineages, some of those will need to be repeated. And in this particular case, the dogs with muscular dystrophy, Andy had mentioned that it's hard to get the gene editing technologies into the right cells, and that's true for muscles, very much so. In this case, though, it only got to about 30% of the cells that they were able to trace, and so, but that was enough that those cells produce enough of the protein to have a restorative or beneficial effect on that disease. So it doesn't have to get into everyone. It's because it is difficult to get them there, and it is context dependent. Now, because they treated just those muscles, it's not going to be passed on to any of their offspring. And most likely, because those edits are permanent at the DNA level, they will that won't have to be repeated. Now, to increase, is it possible to do that again? That research remains to be seen, but in this case, it was only done once.
Okay.
Yeah, those changes will last for the lifetime of that cell. As Casey said, yeah, sometimes certain cell types turn over or die. Their lifespan is very short. In cells or tissues like that, like the blood, the more editing that you can get in, say, blood stem cells, which live in the bone marrow, you can bring them out, the longer that therapy, cure, change will last, because it will last in all of the cells then that those stem cells, those blood stem cells make. If you only do the cells that have a month of life in them, some of the immune cells, then that therapeutic will only last for about that long.
So someone had also asked, how far away are we, do you think, from gene editing becoming a standard of care for certain diseases, and maybe even covered by insurance? Not that you're insurance experts here, but how long until that is sort of seen as a standard of care, or is it already in some cases?
Standard of care. That is a great question. I don't think we're there yet, and I don't have a crystal ball. It's going to be context specific as well. Some will be easier than others, but we're still in the early clinical trial stages. There's been a lot of promise and hope for those, but I think we're still quite a ways off, in my opinion. And time-wise, I don't know. I think, yeah, get context dependent. Some will be easier than others, so they'll be able to get through clinical trials, have success over current standard of care, right? So if gene editing is possible, it's safe, but it doesn't provide any benefit other than what's already out there, then it won't be a standard of care. So it has to show extreme benefit. And I misspelled a word. I think I answered a question in the chat here about CRISPR and the acronym, how it's spelled. And then I wrote it out, but I think I spelled something wrong, interspersed, I think. But so forgive me for that one, but that's what it stands for.
It depends on the type of therapy. It depends on the type of disease. So one of the first gene editing therapeutics that got FDA approval a few years ago, Luxturna, targets a particular gene responsible for a form of inherited blindness in adults. And it opened up with a $2 million price tag. It works. You don't have to edit all of the cells in the retina to improve vision. So then the question became, how will that company work with insurance providers to get that covered? It requires a diagnosis of a very specific genetic mutation that is somewhat rare. So for CRISPR to be used as a standard of care for that particular genetic mutation, that's very possible as long as the insurance companies or the patient has the ability to pay for it. Other therapies, personalized medicine is absolutely the future of medicine. That requires individual sequencing of a person's genome or of a region of their genome that is predicted to be, that contains the mutation or that contains a genetic change that results in the disease. That may be cancer. There are clinical trials ongoing right now for using CRISPR along with another tool called CAR-T, which is a way to modify a person's T cells or their immune cells. We've heard a lot about T cells over the last year with COVID, as your kind of longer term memory immune cells that will go out and really continue to fight the good fight in your body, teaching those cells, giving them the instructions to go and fight a cancer. Now, this has a long way to go because it's not just a quick, easy, clear win in this case. But with cancer, and especially certain types of blood cancers, leukemias, AML, this could become, the goal is for this to become standard of care, for there to be a pipeline where a person will go into the hospital, they will have their immune cells drawn from their blood, cultured, edited, verified as to which ones got the correct edits that will allow that cell to target the cancer, purified, grown up, purified, and then put back in that patient over the course of however many times they need to do it. Maybe it's once over the course of a couple weeks, a week. That has the potential to become a more routine therapy once it's really nailed down.
Oh, that's amazing. I'm going to shift the discussion just slightly to more about plants and crops. I know we've been talking about humans and a little bit of animals. Someone asked, are there organizations working on gene editing in plants or crops to increase productivity, or maybe just kind of broadly what's going on with plant science and gene editing?
I can
start
go ahead.
a little bit. There are plenty of universities, lots of universities around the world who work on this. There are plenty of companies, especially companies here in St. Louis, like Monsanto or Bayer now, smaller startup companies around the country, using gene editing to advance the way, either to drive increased resistance to a particular disease or fungus or insect, or to increase production of something good. So, for example, soybeans. There's a group that's up in University of Minnesota that's been using another one of the gene editing technologies called TALENs to change the genome or to create mutations in the genome that allow soy to produce more healthy oils so that we can harvest soy for more health that allow the plant to make more of particular types of soy oils that can be used then for healthier frying, for healthier cooking. That's one example, is you can allow a plant to produce more or less of something that allows it to be healthier or last longer on the shelf. Genetics in plants and genetic manipulation of plants has been happening for a very long time. for a very long time.
Yeah, I think I would add. Oh,
No, go ahead.
just add to that. That yeah, I think the tools for gene editing in crops and I would even say synthetic biology, microbiome, micro organisms to produce, things like fuels and different cosmetics, for instance, and different things that we as consumers take in. With plant breeding, this is just a more sophisticated tool to grow those crops so that they can grow on, in arid conditions, grow faster, and where in climates where they don't get as much sun, low, not poor soil conditions. And I think that's where we're going to see a lot of advances. And then also the microbiome for those plants to grow. Those can be edited as well, and that doesn't necessarily affect the plant and its genome. So I think there's a lot of promise there to help, feed the world as we have growing populations.
There's been a lot of, I guess, bad marketing, really, from scientists and from policymakers around genome engineered or genetically modified crops and organisms. In part, many of the early genetically modified crops or livestock used a gene used a gene from a different animal or from a different plant or from a different species to better perform a function inside of a different organism. So that would be, let's say in some salmon, that would be taking a gene or from another larger species of fish, replacing a gene or putting it into salmon that allowed them to be grown to grow larger. That's called transgenesis, or a transgene, where you're taking a gene from a different organism and putting it into a different organism and or a different species, and allowing that to gain better gene function, better physiological function of that organism, that plant. The PR around genetically modified organisms has caused a lot of, problems, a lot of confusion around the safety of this particular type of technology in something like food, in food, for instance. With gene editing using something like CRISPR or TALENs or zinc fingers, you are modifying you're modifying the genome, making changes to the genome to basically do the same type of thing without putting in DNA sequences or proteins from a different organism in. And from a communications or from maybe a marketing or a policy standpoint, that seems to be a much easier, a much more acceptable way to modify the genome. Plants have been modified for a long time in very non-specific ways. We expose plants to X-rays that allows them, and then we expose 1 million seeds to X-rays, which is going to create random damage in the genome. And then we plant all of those seeds and look to see which one grows up and does the thing that we're looking for: a taller plant, a plant that makes more kernels of corn or more ears of corn per stalk. It's a random mutagenesis. It really is just a speeding up of natural evolution, but it's a lot of work to do that. It costs a lot of money and it's a lot of work, and it's safe. It's just a lot of work and a lot of time. Gene editing using CRISPR or TALENs or zinc fingers allows us to get to that place much more efficiently, where we can make specific changes, or we actually know what we're doing to the genome to allow us to get crops that are better, healthier, grow stronger, grow in places where they can't grow otherwise.
Oh, that's great. Thank you for that description. We have one more question from the audience, and then we'll have one final question after that. This kind of goes back to an earlier discussion, but is it possible, you think, in the future to treat an autoimmune disease that is inherited using gene editing, assuming they could find the error in the genome?
Okay.
Yeah, sure. I'm not an autoimmune expert. I don't know if they'll find a gene. I think it depends on a lot of factors, and could be multigenic, if you will. I think a lot of the examples, like sickle cell anemia, let's see, what's another one? Cystic fibrosis, Duchenne's muscular dystrophy. These are single genes, monogenic genes that are identified as, causative for this whatever particular disease it is. There are genes that may have just a background that predispose you towards, developing autoimmune disease. So they're very complex. But I would hope that, given that we have these tools, like I said a little bit earlier, we have the ability now to look at all of the genome and very, powerful experiments that even go down to the single cell resolution, looking at the genomes and how they're affected by different, modulators. I hope someday we will figure out the mechanisms about immune diseases. And, I don't know whether CRISPR will be the tool that can help, but I definitely think it's not within, it's within reach possibly to be able to discover those mechanisms, which I think are very complicated. So I think that was a really good question. It's not always super easy to say, oh, this is it. If we just do this, it's fixed. But
we have a better chance for something like that because it's your immune system is generally bloodborne, and so the ability to make changes to blood lineages is a lot easier than you can replace your blood. You can replace your blood stem cells. You can't replace your parts of your brain. You can't replace other parts of other organs as easily as you can with blood. So again, I'm also not an immunologist, but the ability to make changes that affect disorders of the immune system are definitely places that are easier to access. So it's not a matter of access at this point. It's a matter of understanding of what's actually going on.
Very cool. Thank you. So one last question for both of you to kind of set our sights on the future. What are each of you most excited about, or what's the thing that you're currently most excited about for the future of gene editing, and what you're working on? And Casey, we'll start with you.
Sure. Oh man, there are so many things that are exciting about. I think genetics is fascinating. I love the work I do, and currently, I guess there's really future of like what's next after CRISPR. CRISPR is so powerful. It's only been around 10 years. What's next after that? But I think right now, what fascinates me the most is understanding the entire genome and the ability to access. Andy mentioned some other tools previously, like TALENs. Those are gene editing tools that are scalpels just like CRISPR, but CRISPR allows you to do so many more types of experiments, ask different questions in different contexts, to be able to understand biology at a much larger scale. So that's what I'm excited for, what that's going to lead to, and the discovery. What else does the genome do? There's so many things that we don't understand yet, and so that's what I'm excited about. But obviously, what grabs attention and headlines is the therapeutic aspect, which is also incredible. So that's my answer.
Good one. Yeah, the ability to take CRISPR, we like to think of that as many people in many of the parts of the discussion we've had today, that's the therapeutic itself is CRISPR. We're putting that inside of a person, but that's only one thing that CRISPR can do from a basic science or from a translational or clinical or pharmaceutical perspective. Using CRISPR to create new different therapeutics is a very hot area. So in my lab, Casey and I, we were working together to refine CRISPR and make CRISPR tools that would be useful for drug discovery. So in a genomics context, and functional genomics is figuring out how the genome works. It is large, it's complex, we don't know everything about it. So using CRISPR to improve our understanding of the genome itself and how the genome works, doing large screens for drug discovery where we independently create mutations in one mutation in each cell, and every cell over millions and millions of cells gets a different mutation, and we observe how those cells behave in response to different drugs. So we can mutate every gene in the genome at once, and then apply a screen of all of the different available FDA-approved drugs and say which one of these genes, if we can tweak this gene, does it make this drug work really well for this particular phenotype or for this particular cellular effect? Or we know that this is the gene that's the most important for this disease. Through that, now we can develop a drug that specifically targets that gene for that disease. CRISPR, not just as a therapeutic, but CRISPR as the tool to discover and refine chemical therapeutics as well. So it's really the key to unlocking a lot of human health. A key.
That's wonderful. Well, we've come to the end of our time. I do want to thank you both again for joining us. This was wonderful information. We will definitely have you back again for something in the future, I am sure.
That'd be great.
And folks who have joined, if you have more questions, I'm going to throw my, I know we didn't get to one of these. I'm going to throw my email in the chat. Feel free to email me and I'll pass along any other questions maybe you think of later, if you two do not mind answering them via email.
Absolutely.
And there you go. Feel free to email me. It should have popped up there. And thanks to our members for joining this evening. And hopefully you all learned something and got to think a little bit more about this topic. We'll see you again the next time we do this. Thank you.
Thank you, Christina. Thank you, Casey. Thank you all for your interest.
Yeah, thank you, everyone.
Have a good evening.